Short questions and answers. Metabolism in enzymes. 1. What is metabolism? Metabolism is the sum of all chemical reactions going on in a cell. 2. Why are enzymes important for cellular reactions? Enzymes speed up biochemical reactions so that their products can be utilized in various life activities. 3. What are enzymes? Enzymes are biological polymers that catalyze biochemical reactions without being consumed. 4. In what form do enzymes exist in the cell? Enzymes exist as colloids in the cell. Structure of enzymes. 5. What type of proteins are enzymes? Enzymes are globular proteins. 6. What are ribozymes? Ribozymes are RNA molecules that act as enzymes, such as peptidyl transferase. 7. What is the function of peptidyl transferase? Peptidyl transferase forms peptide bonds during protein synthesis. 8. What forms the three-dimensional structure of an enzyme? A linear chain of amino acids forms the three-dimensional structure of an enzyme. Active site of an enzyme. 9. What is an active site? An active site is a specific charge-bearing, three-dimensional cavity where the substrate binds. 10. What are substrates? Substrates are reactants that are converted into products in enzyme-catalyzed reactions. 11. What types of interactions attach substrates to the active site? Non-covalent interactions, such as hydrogen bonding and hydrophobic interactions, attach substrates to the active site. 12. How many amino acids form an active site? An active site typically consists of 3 to 12 amino acids. 13. Which amino acids form the active site of aldolase? Glycine, histidine, and alanine form the active site of aldolase. 14. What are the two functional regions of an active site? The two functional regions are the binding site and the catalytic site. 15. What is the function of the binding site? The binding site binds the substrate to the enzyme. 16. What is the function of the catalytic site? The catalytic site converts the substrate into the product. 17. How does the active site's shape relate to the substrate? The shape of the active site is specific to the substrate, allowing only a particular substrate to bind. Cofactors and enzymes. 18. What are cofactors? Cofactors are non-protein components that help enzymes in catalytic processes. 19. What is a holoenzyme? A holoenzyme is an active enzyme formed when a cofactor combines with an apoenzyme. 20. What is an apoenzyme? An apoenzyme is the inactive protein part of an enzyme without its cofactor. 21. What is an example of an enzyme that can exist in active and inactive states without a cofactor? Pepsin is an example of such an enzyme. 22. How is pepsinogen activated? Pepsinogen is activated when exposed to HCl, which removes an additional polypeptide fragment. Types of cofactors. 23. What are the three types of cofactors? The three types are inorganic ions, organic molecules, and prosthetic groups. 24. What are inorganic cofactors? Inorganic cofactors are metallic ions like iron, magnesium, copper, and zinc that are detachable. 25. What is an example of a reaction involving an inorganic cofactor? Glucose plus ATP, glucose 6-phosphate plus ADP, with hexokinase plus magnesium. 26. What are organic cofactors? Organic cofactors are coenzymes or prosthetic groups, often derived from vitamins. 27. What are coenzymes? Coenzymes are organic molecules that detach after the reaction, like NAD and FAD. 28. What is a prosthetic group? A prosthetic group is a permanently attached, covalently bonded part of an enzyme. 29. What is an example of a prosthetic group? An iron-containing porphyrin ring in cytochromes is a prosthetic. 30. What is an example of a reaction involving a coenzyme, ethyl alcohol plus NAD plus acetaldehyde plus NADH2, with alcohol dehydrogenase?